أهلا بيكم في دروس التشريح دروس الأحياء للصف الثالث ثانوي لغات. Today's lecture will start a new topic, which is nucleic acids and protein synthesis. So let's begin. The structure and synthesis of proteins. Proteins, you know, are essential for control of different parts of the body of the living organisms, you know, such as enzymes and hormones and antibodies, etc. So they are needed. To understand how they are synthesized, we must take a look at the structure of proteins. They are the building materials in living organisms, and there are, you know, the fibrous proteins that take place in the form of actin and myosin of muscles, or collagen fibers all over the body, or keratin layer that forms covering the skin or hair. All these are made up of proteins, and those are called structural proteins. Another type of proteins, which is regulatory proteins, I told you about how the body needs enzymes and antibodies, hormones, and various other substances that make the living organism respond properly to the changes in his environment. So they are essential too. There are 20 amino acids involved in formation of proteins. They have the same basic structure, which is a carboxyl group, amino group, and a hydrogen ion, okay? And an R group, except in glycine. Glycine has another H here to make the glycine amino acid. So all the others have the R group we're going to talk about now. See, except for glycine, all the rest have the R group, and amino acids are joined together by peptide bonds to form polypeptide chains. The differences between different proteins are due to the following: either the number of the amino acids, change in the kind of amino acids, or arrangement in amino acid sequence. This also makes a difference, and the number of polymers that form the protein. So the major differences between proteins come from differences in numbers, differences in kind, differences in arrangement of amino acids in polymers, and differences in number of polymers that form the protein. All these are joined by hydrogen bonds that may give the protein molecule its special shape. Ribonucleic acids. We studied DNA, which is deoxyribonucleic acids. This time we're going to study ribonucleic acids. They are the same like DNA, macromolecules made up of nucleotide subunits, and each nucleotide, like DNA, is made up of sugar molecule, a nitrogenous base, and a phosphate group. The phosphate group is bonded to the third carbon of the sugar of the nucleotide next to it to form the sugar phosphate backbone. All this was studied with the DNA, but there are differences between RNA and DNA. For example, the sugar in the RNA is ribose, not deoxyribose. That's why the DNA is called deoxyribonucleic acid, and RNA is ribonucleic acid. RNA is usually a single strand, and the DNA is a double strand, but you can get double strands, complementary chains of nucleotides. DNA differ from RNA in the kinds of nitrogenous bases they contain. I mean, both of them contain adenine, guanine, and cytosine. But in DNA, it contains thiamine, and instead of thiamine in RNA, it's replaced by uracil. And also uracil, like thiamine, bonds or base pair with adenine. Okay, so another difference between RNA and DNA: adenine, guanine, cytosine present in both. But instead of thiamine of DNA, there is uracil in RNA. Okay, to study RNA, three main types of RNA participate in protein synthesis. They are the messenger RNA, ribosomal RNA, and the transfer RNA. The messenger RNA transcription begins in it when the enzyme RNA polymerase binds to a sequence of nucleotides of the DNA strand. This sequence of nucleotides is called promoter. So the RNA polymerase binds to a promoter, which is a certain sequence on the DNA strand. Now, two strands of DNA are separated, and RNA is being a complementary strand formed on one of the DNA strands. Okay, the enzyme works, the RNA polymerase works in the direction we're used to, that is 5 to 3, but it's in the one it's synthesizing. 5 to 3 in the RNA it's synthesizing. 3 to 5 of the template, of the DNA template. Okay, and this makes sense. We understand this, we studied this in the past lecture. So this is the action of RNA polymerase. It's like the replication of DNA, but we're used in replication of DNA that it involves all the DNA molecule. But in RNA, it's only a selected portion of the DNA. Okay? Only one of the strands is transcribed in one segment of the DNA. The orientation of the promoter indicates which strand is to be transcribed. The difference between eukaryotes and prokaryotes is that prokaryotes, there is a single RNA polymerase that transcribes the rRNA, mRNA, and tRNA. But in eukaryotes, there are 3 different kinds of polymerases. So this is the difference between prokaryotes and eukaryotes regarding the RNA polymerase. Okay. In prokaryotes also, it's the RNA, the messenger RNA is ready for translation as soon as it's transcribed. I mean, it can bind to ribosome at one end and the other end is still being formed. Okay, there is a binding site for the ribosome on one end, as you can see here, ribosome binding site, and it has a certain sequence which is AUG. And there's, after a while, there's a stop codon, which is one of 3, UAA or UAG or UGA. تمام. And the other end of the mRNA molecule في حاجة اسمها polyadenine tail زي ما انتم شايفين هنا حوالي 200 adenine residues. Discovered that these adenine residues are responsible for protection of mRNA from breakdown by enzymes in the cytoplasm. اوكي، يبقى بيحموا الام ار ان اي من انه يبقى يتكسر بالانزيمز. زي ما قلت لكم في البروكاريوتس ان ممكن ان بدايه الام ار ان اي تبقى اتاش للرايبوزوم والاذر اند لسه بتتعمل في دي ان اي تمبلت. لكن في اليوكاريوتس لازم الام ار ان اي لازم يبقى processed further in the nucleus before the mature mRNA is ready to enter the cytoplasm and participate in protein synthesis. يبقى دي فروقات بين البروكاريوتس واليوكاريوتس. اوكي، قلنا في البروكاريوتس a single RNA polymerase transcribes R و M و T RNA، لكن 3 different kinds في اليوكاريوتس. وقلنا كمان ان في حاجه اسمها ribosomal binding site و polyadenine tail at the end that protects the mRNA from breaking down by enzyme. وقلنا كمان من الفروقات ان ممكن في البروكاريوتس الام ار ان اي يبقى مرتبط من ناحيه بالرايبوزوم والناحيه الثانيه لسه بتتعمل على دي ان اي تمبلت. لكن في اليوكاريوتس الحل لا، لازم الام ار ان اي يبقى جاهز must be processed further in the nucleus before the mature mRNA is ready to enter the cytoplasm and participate in protein synthesis. So remember the ribosomal binding site at one end, polyadenine tail at the other end, the start codon which is AUG, the stop codon which is one of 3 codons. Ribosomal RNA, ribosomes consists of several types of RNA, about 70 kinds of polypeptides. In eukaryotes, ribosomes are produced in the nucleolus, which is inside the nucleus. With several hundred thousands of ribosomes are produced per hour. And to explain why the number is that fast in such a short time, because the answer is, like we are used to, is the number of copies of ribosomal RNA genes is around 600. So that's why when you repeat a number of genes, copies of genes, means that it's a trait and it will be more pronounced. So the cell makes a lot of rRNA is transcribed. There are 4 different kinds of rRNA that participate with the protein in the ribosome structure. Ribosome consists of 2 subunits, a large one and a small one. And they when ribosome is not engaged in protein synthesis, the 2 subunits wander independently, and they may have join different subunit of the opposite type the next time they participate in protein synthesis. Okay. In eukaryotes, also the difference between eukaryotes and prokaryotes, polypeptides of ribosomes are made in the cytoplasm, then pass through the envelope of the nucleus and enter the nucleolus, where the rRNA and polypeptides are assembled into ribosomal subunits. So this happens in the nucleolus, but the polypeptides are made in the cytoplasm. Please pay attention to this. The polypeptides of the ribosomal subunits are made in the cytoplasm. They are carried inside the nucleus to the region of the nucleolus, where the rRNA and polypeptides are assembled into ribosomal subunits. The transfer RNA, it's a 3rd kind of RNA that participates in the protein synthesis, which is the transferred RNA. For each kind of amino acid, there is a tRNA specific to it that recognizes to it and attaches to it. Transfer RNA is transcribed from tRNA genes arranged in clusters of 7 or 8 tRNA genes in the same part of the DNA molecule. As you can see here, this is the shape of the tRNA molecule. It has a peculiar shape where the sequences throw themselves into loops at multiple ends, as you can see here. There's two important sites on the tRNA. This site, which starts with the codon ACC at the 3 end of the molecule. This is the amino acid where the amino acid attaches. So this is the site where amino acid attaches to the tRNA molecule. So as the tRNA molecule takes it to the mRNA and starts its work. So to bind to the mRNA, there is another end called the anticodon site to bind with the mRNA, appropriate mRNA codon at the mRNA ribosome complex. Okay, these codes makes the mRNA, the tRNA, able to put the desired amino acid in its correct place, in its appropriate place in the protein. So this is the transfer RNA, or the third kind of RNA. This is a hint about the structure of RNA. And next time we will continue our talk about ribonucleic acids. I hope you got the maximum benefit from this lecture, and thank you very much for watching.